where it builds up is what we don't know. But, all right, cool, Susan. So this is getting more into health-related effects. In the Microbiology of the Built Environment program for the Sloan Foundation, we were always told not to focus on health effects, but it's become increasingly apparent that the only relevance is really health effects because there's not much ecology.
I would say it's not the only relevance, but it at least gives us an objective marker of the status of the microbiome of the built environment. So what I want to spend the next 15 minutes or so going through are some of the studies that we've been engaged in relating the microbiome of the built environment specifically to childhood allergic asthma. And just to begin with, to give you some kind of framework for this, we kind of know that allergic asthma is a disease that is really considered to be driven by environmental factors. And this is largely because though we have some risk alleles and combinations of risk alleles that increase susceptibility, the prevalence of the disease has increased so dramatically over the last several decades that it just cannot be down singularly to genetics. And these are stratified basically by the age of the participants. And you can see this red line here is the prevalence of allergic asthma in children. And it has been rising steadily over the last several decades. It's reached an asymptote over the last decade or so, but it's been rising. And the other key player here is that the distribution of the disease is really quite skewed towards that of developing nations and kind of the Americas, Australia, and pockets of Western Europe, where lifestyle is quite different compared to that of developing nations. This is where we see the burden of disease to be greatest. And some of the wonderful work by Erika von Muchus and her group have shown that living on a farm and early life exposure to livestock is actually protective against allergic asthma. And you can see these are from two very large independent studies that were put together for a New England Journal of Medicine paper. And what you can see is that the prevalence of the disease, either asthma or allergic disease, or atopy as we call it, is significantly reduced in children who grow up in early life in farming environments. And what they went on to show in the farming environment is that these children who grow up on farms are exposed to a greater burden and numbers of distinct bacteria and fungi, and that as this increases, these microbial exposures increase, the prevalence of asthma decreases in these populations. So exposure to microbes in early life is protective against allergic asthma development. And we came into this thinking microbiologically about this conundrum. And there's been a lot of epidemiological data in the field that's been really validated across large numbers of studies. And what we came upon was that the risk factors for the development of childhood allergic asthma really are factors that we perceive as having a strong selective pressure on the gut microbiome, and the airway microbiome, on the developing microbiome in very, very early life. And they include, as I've just shown, exposure to livestock or to furred pets, formula feeding, which we know skews the microbiome towards that which is deficient of bifidobacteria, cesarean section delivery, which Maria Gloria Bello Dominguez has shown quite beautifully, results in very distinct early pioneer organisms at a variety of sites in the neonate, and maternal antimicrobial exposure during pregnancy or early life antimicrobial exposure. These are all risk factors for developing the disease. So what happens in very early life? This is from one of our studies, as yet unpublished. These are stool samples collected from 25 children. This is meconium. This is the first bowel movement of these children. And we can see a breadth of bacterial diversity in this first stool sample. We don't know whether it's live or dead, but there's signatures of bacteria present. Irrespective of this breadth of diversity in meconium, all children develop bacterial diversity expansion over the first year of life. This is samples collected at 1, 3, 6, 9, and 12 months of age. And it's really compelling to us that many of the risk factors for the development of childhood allergic asthma are really centered around this critical period of prenatal and very early life. And the kind of evidence from culture-dependent studies had indicated that the gut microbiome may be a good place to look for factors that may dictate susceptibility to develop disease. And two independent studies from John Pender's group and Kaliomaki and colleagues showed that children who are 3 weeks of age, who had stool samples that were enriched for either Clostridium difficile or Escherichia coli, were at a significantly higher risk of developing allergic asthma. So we asked the question, where does these microbes that populate the developing gut microbiome in very early life come from? And in Western nations where the disease burden is highest, we spend an inordinate amount of time in the built environment, and particularly in our own residences. So putting all these pieces of the puzzle together, we came up with a hypothesized causal pathway of childhood allergic asthma in which the built environment, and specifically the microbes in the built environment, influence the very early life developing microbiome. We focused on the gut microbiome, but that's not to say that this doesn't happen at other mucosal sites. And that microbiome, in turn, influences the immune response. This is a two-way conversation because this feeds back upon the microbiome and its constituent membership. And that ultimately these components dictate the susceptibility to disease development. So if this is true, you might imagine that the bookends of this causal hypothesis may hold out. And so we hypothesized that early life house dust microbial exposures are related to allergic disease, atopy, and or allergic wheeze development in childhood. And so one of the first studies we did in this arena was with the Inner City Asthma Consortium, which is led by Bill Bussey. And we leveraged samples collected by Jim Gurn from the Eureka, or Urban Environment and Childhood Asthma Study. And this is a prospective cohort study of full-term infants in inner city urban US neighborhoods where the burden of disease is quite high. Jim and his colleagues had enrolled 609 families across 4 different cities, and they had collected house dust at 3 months of age, when children in those homes were 3 months of age, and then they had defined atopy, or the propensity to develop allergies, at age 3, and the propensity of these children to be recurrent wheezers, which is often seen as a precursor to developing asthma later in childhood. And so we took a subset of 104 samples, house dust samples, from this cohort, for which there were 3-year atopy and recurrent wheeze data available. And they parsed out into 4 relatively equally numbered groups, where we had healthy children that were neither atopic nor recurrent wheezers, those that were simply wheezers, those that were just atopic or allergic, and those that had both atopy and wheeze. And what we found was that when we profiled the bacteria present and calculated these kind of gross indices of community summary statistics, essentially, for these communities, community richness, the number of types of bacteria we detected, evenness, their distribution within the communities, and diversity. Here we use the Shannon index, which is really these 2 indices combined. What we see is that as we go from the healthy children through the wheezers, the atopics, and the atopic wheezers, we lose bacterial richness. They become far more uneven communities, and they are less diverse. We did some comparisons across these groups and asked, well, which are the organisms that we find to be enriched in homes where children do not develop disease? These are protective bacteria. Or potentially protective bacteria. And we have a long list, I'm not going to call out the names of them, but what was really interesting is when we compared the neither versus the atopic, or the neither versus both, there was a subset of organisms, shown here in the purple, that we consistently find to be significantly enriched in homes in which children do not develop allergic asthma. And this was really an echo of an earlier study we'd performed, and it was based on an observation by Christine Cole-Johnson and Dennis Ownby, made in a publication in JAMA in 2002. And so they were the ones from their epidemiological studies that had shown that children exposed to dogs, and to a lesser extent to cats in early life, were protected against childhood allergic asthma development. So we hypothesized that, again, there's a microbial signature that goes along with those kind of pet-associated exposures in the home in early life. So we took a number of house dust samples from homes with dogs, cats, and no pets present, which run a difference in their protection against atopy development in childhood. And to summarize what we found that in the homes there were dogs present and had the highest protection against allergic disease, those homes had the greatest bacterial diversity and the lowest fungal richness. And that this kind of ran a gradient as we went through these samples, in that those homes in which there were no pets present had the lowest bacterial diversity and the highest fungal richness. And I should say that this is quite distinct from what we see in farms in Bavaria, because these are not farms in Bavaria. These are homes in urban areas and rural areas around Detroit. And indeed, the devil's in the details, because these fungi that we identified were highly allergenic, as Cladosporium, Aspergillus, so that really we need to get down to the specifics of these exposures in these homes. But that was great. So there's a microbial dichotomy in the exposures of children who do or do not go on to develop allergic disease in childhood. But the next question was, do these exposures actually influence the gut microbiome in any meaningful manner, and especially in a manner that influences immunity of the mucosal surface of the airways? And so we collected house dust from homes with dogs and no pets present, which we saw as the kind of veritable bacterial desert. And we profiled them, and we ran a little experiment with mice. We took mice and we fed them these kind of very microbially distinct house dusts for 7 days as an oral gavage. So we're really trying to recreate what children do in their homes. They go hand-to-mouth behavior. They suck in what's in the house dust on the floor. So 7 days of exposure, and then we continued to expose the mice twice per week to keep up that exposure while we sensitize their airways to cockroach allergen. And we do this simply by adding cockroach allergen to their airways. And we need to do this 3 times over a 3-week period to sensitize the airways. We ended the protocol and took down the mice. And what we found was quite stunning. We found that only the animals who had been supplemented with house dust from the dog-owning homes showed significant reductions in allergy-associated cytokines, IL-4 and IL-13. This didn't occur in the mice that had the house dust supplemented from the homes with no pets present. We saw that mucin secretion genes were, again, only significantly reduced in those animals that received the dog-associated house dust. And this is the histology, a picture's worth 1,000 words. These are control animals. These are their air spaces. This is the airways of these animals. So here are the controls. They never saw any supplement. They never saw any allergen. Their air spaces are pristine. These are the animals that we sensitized. And now you see that the air spaces is now occluded with pink staining mucin. There's a lot of nucleated cells around here. It's an inflamed airspace. These are the animals that received the no pet associated house dust. Again, there's a lot of mucin in these air spaces and inflammatory infiltrate. And these are the animals these are the mice that received the dog associated house dust. And I would counter that these air spaces are akin to the sensitized animals in that they are equally pristine. So remember, the idea was that this actually occurs through changes in the gut microbiome of those animals. And so we went on to profile the cecal communities, the bacteria in the cecal communities of these animals. This is a principal components analysis. Each spot represents a bacterial profile in a single animal. We've colored them by the group that the animals are in. These are control animals. And you can see that when we supplement the animals with house dust, we shift them. There's distance between this group and the other 2 groups. And that indicates that we have changed the bacterial community profile in those animals to a significant extent, although I don't have the p-value up here. So we significantly changed the composition of the gut microbiome of these animals when we supplement them with house dust. But that's really great. That's all in mice. But I'm really interested in understanding how we can prevent allergic asthma development in children. And so we asked whether something similar occurs in children using another birth cohort. This is Christine Cole-Johnson and Dennis Ownby's birth cohort from the Wheels birth cohort, which is centered around the Detroit metropolitan area. And these studies in my group were performed by Kei Fujimura, a wonderful staff scientist that I've worked with for years. So the birth cohort is a socioeconomically and racially diverse population. We just took a subset. There's about 1,000 children in this study. We took 300 stool samples, and these ran the gamut across the age of these children. Some of them were very, very young, just very close to birth, and others were 11 months old. These are all independent samples. This is not a longitudinal study. And these were really, when the guys collected these stool samples, they were really targeting 2 kind of age groups. One, a neonatal age group, and the majority of those samples, 130 of them, are around this median age of 35 days, so very, very young samples. And the remaining 168 samples that we examined are much older. They're about 201 days of age as the median age, and there was a much larger range in the infant population that we studied. We started by just asking what the bacterial and the fungal communities looked like in these stool samples using sequencing. And rather than stratifying these children by their clinical outcomes, we took a different approach to this. We know that microbes have developed a wonderful biochemical lexicon to interact with each other. And this dictates both the burden and the activity of organisms in microbial communities. So we came up with the idea that there may actually be, rather than one type of microbiome, there may actually be compositionally distinct microbial states in very early life that actually interact differentially with the host and dictate susceptibility to allergic asthma development. And so, in the interest of time, this is the neonatal data that we found. We found 2 different microbial community states in the infants. We found 3 in the neonates. And this is not an age-based stratification. There's no significant difference in the age of these participants. And what we see is again, this is one of these PCOAs. All the blue dots are one group. We call this NGM2. It was typically Enterobacteriaceae dominated. NGM1 is this middle red group. They are dominated by bifidos. And then NGM3 is this kind of mixed, strange group that was not dominated by one organism, but actually by 2, both bifidobacteria and enterobacteria. And this explained about 9% of the variation in community composition, these kind of stratifications based on this compositionally different states. The next thing is whether these states are actually related to the propensity to develop disease. For the infant gut microbiota states that we found, there's no significant difference in relative risk of either atopy or parental reported doctor-diagnosed asthma. But when we move to the neonatal group, what we find is that NGM3, this third and kind of strange community state that we found, runs a significantly higher risk of developing allergic atopy compared to NGM1 or NGM2, and a significantly higher risk of developing doctor-diagnosed asthma at age 4. These are interesting communities because NGM3 lacks many of the bacteria that we associate with beneficial effects, and many of the organisms that we've actually found to be enriched in the house dust of homes where children do not develop allergic asthma. Even though these are very different studies in independent cohorts, the key organisms that we find here, Akkermansia, Faecalibacterium, and Lactobacillus, are amongst those that are most highly enriched in the homes in the other Eureka study that are associated with protection against allergic asthma. But what was stunning to us was that these children who have the highest risk of developing allergic asthma are really highly enriched in Candida and other fungi. And this has not been shown before, so it's not just about bacteria. There's an interkingdom dysbiosis in the very early gut microbiome of children who go on to develop allergic asthma. So this is all great. We have these parts of the components in humans. We know now that disease susceptibility is related to microbiome composition, and we have some data, though I haven't shown it, showing that it's related to the immune response of those children. But what about the microbial environment, the homes in which those children have been raised? Well, with the great fortune that Chris had collected actually house dust samples from these children in 2002 and onwards, and 120 of the samples for which we developed a gut microbiome profile actually had a paired house dust sample available as well. So we performed the exact same profiling with the exact same primers. These are all approximations of which microbes are present. And we're really interested in understanding which variables that have been collected by Chris and her colleagues actually explain variation in house dust microbiome composition. And in particular, was there any relationship between the type of microbial state that a child has who's raised in those households, which dictates a susceptibility to disease, and the type of microbial exposures they have in early life? And so it was very gratifying to hear many of the earlier presentations, and especially Brent's, of there's a large number, this is a tiny subset of what we found, but what we're finding is very similar to other studies. There's a large number of variables that have very small effect sizes on the built environment, bacterial and fungal communities. But what keeps coming up is that occupancy, and in our studies, ethnicity and smoking are some of the factors that are co-vary with variability in bacterial community composition. And what was very exciting for us is that the type of neonatal gut microbiome a child has in very early life is related to the bacterial exposures in their homes. For fungi, what we find is that, again, demographic and environmental factors such as exposure to dog in very early life are related to the types of fungi that are present in these homes. And again, even more exciting, not only is the type of neonatal gut microbiome associated with the fungal community in the home, but the propensity to develop atopy against peanut, it's actually against other food allergens, is also related to the types of fungi that are associated with these homes and the types of fungal exposures that these children have in very early life. So we finally have some very nice evidence that the type of neonatal gut microbiome is related to disease susceptibility, and it is also related on the other bookend of the causal pathway to the types of microbial exposures that those children have in very early life in those homes. So where do we go from here? I think we have to move away from taxonomy. We have to move away from the packaging that's around these microbial genomes and move towards understanding the actual functions that are transferred from the built environment to the human host that impact health status of occupants in the built environment. And I think this is a moving target as well. I don't think that there's one answer to this. I think we need to start beginning to understand whether the relationship between the microbial built environment and the host really differs with age, with gender, with health status, and an anatomical site, the skin versus the gut versus the airways. And I think there's a large amount of work to be done in this realm, and until we start to really disentangle these kinds of effects that have, these kinds of variables that have small effect sizes, but cumulatively may have a large propensity to act as biomarkers for the interactome between the host and the human host and their built environment. I think this is what will propel us towards understanding what are the steps we can take to modify the built environment to promote host health. And clearly, this work took a very large team of people, not least Homer Boucher and Kei Fujimura at UCSF, and Chris Johnson, Ed Zarate, and Al Levin at Henry Ford Hospital, Dennis Ownby, who's now at Georgia Health Sciences University, and Nick Lucas and Tina DeMoore, who helped us with the mouse studies, and of course the NIH and the Sloan Foundation, who have funded us for many years. I'm happy to take any questions.
I think your work is so awesome. So question for you, I often get asked what the relative importance of, in terms of a microbiome source, that the built environment versus food might be. And so how do you have thoughts about how to disentangle, I mean, is it possible that the microbiome in the gut, for example, in your studies is really a function of some other source other than the built environment?
Yeah, you can see from the kind of the effect sizes, the R squared is 2% of the variability in the house dust fungal microbiome, for example, is related to the neonatal gut microbiome state that we see. So yes, it's a confluence of influences on those. You know, feeding practices, obviously, these are formula or breastfed children, will influence, we think, the composition of the community that's present. And we think that the composition of the community may play a large role in dictating the interaction of that child with the microbiome in their built environment. That some communities may be more receptive to being colonized by environmental microbial exposures, while others, which drive a pro-inflammatory state, would be a more hostile community or ecosystem for environmental microbes to actually colonize. So this is something that we're thinking in terms of and working on now. Yes.
Early life exposure is very important, as you've demonstrated here. Do you have any sense as to how quickly that window closes and why it closes, and how amenable to manipulation that window might be at later stages in life?
That's a really good question. I think the main things we can point to is what we know about gut microbiome development in early life. And the general dogma is that somewhere between 1 and 3 years of age, that at least at the phylum level, the distributions are relatively set. My sense is that if there's a perturbation, there's now open space for colonization. So and that can happen at any stage of life, you know, in later life and adulthood. So there may be periods where for example, the gut microbiome or the airway microbiome may be more amenable to colonization from the environment. But the general sense is that it's that critical period in the first year of life that matters immensely for a colonization from the local environment and other sources, as Jessica pointed out. But my sense is if there are perturbations, that opens up space for colonization, and that could allow for environmental organisms to colonize also.
Yeah, Susan, yeah. So very nice talk. So I saw you have one slide. This is the R square value and the P value. So in different conditions. So all those R square, the P value very significant, less than important than the other one. But if you look at the R square,
They're tiny.
very small.
Yeah,
So altogether only like you explain 80%.
Right.
What other 90% of very? How can you explain this?
Well, is it that there's a 90%, or is that when you add those variables together, you come up to a cumulative influence on the microbial exposure in the home? So this is all univariate analysis. And so one of the things we've been thinking of, as I brought up earlier this morning in one of the earlier talks, is if we use multivariate models, do we see combinations of variables that explain a greater proportion of variance in those communities, in those populations? So that's now what we're doing with this data. This was just literally a first pass. The other obvious thing is we haven't measured what these were what most influences these communities. These were questionnaires that were developed, 10, 12 years ago, not thinking in terms of the microbiome and its variance, but thinking in terms of trying to capture as much information as possible in an epidemiological study. So they may not be ideal. We have actually, with Sloan funding, collected another 100 samples from another 100 homes, and we've worked with Stuart Batterman, a building scientist, to develop a much more robust questionnaire to kind of have a greater assessment of kind of structural features of the building. And perhaps some of those variables may explain a greater proportion of the variance in community composition.
Okay, thank you.
And that questionnaire, sorry, I think that'd be a really useful thing to have. I don't want to steal something that you're producing, but if that's something that's being developed, will you make that publicly accessible?
Yeah, sure. As soon as we get some work out from it, we need to find funding to actually analyze those samples. So, but yeah, this is something we worked hard with Stuart. Be happy to share it.
Have you been trying to look at the mechanisms whereby children on farms versus children with pets have clearer respiratory passages than ones who have not had animals?
We have, and I didn't really have the time to get into this. So that study of the neonatal gut microbiomes, we've driven much deeper with that. We hypothesize because these are compositionally distinct communities that they're also functionally distinct and metabolically distinct. And so there's a growing body of evidence that it's called immunometabolism, that the metabolic state of the cell dictates its phenotype. And so we've come up with the idea that the metabolic microenvironment that host cells see or sense is dictated by the types of microbes that are there and what they present to the host. So we went on to show that the three distinct neonatal gut microbiota are basically associated with very distinct metabolic microenvironments. And if we take sterile stool water from representative samples from those children and run an assay with dendritic cells and T cells, we can show that the metabolic microenvironment in that highest risk group drives an allergic response. So these children actually have an allergic response, we think, develop very early in the gastrointestinal tract, and that this is what precedes the development of allergic disease and asthma, and the manifestation of the disease in the airways, we think, comes later. We further even gone into it to identify specific metabolites that drive the immune dysfunction associated with allergic asthma in those children. And so now we're working back to understand who are the microbes in those dysbiotic communities that have a greater propensity for developing disease that produce those metabolites. So trying to, we work forward and then we work back. We try to understand an outcome and then we try to pull it back to understand what drives that outcome at various levels, either at the microbe level or the built environment level.
One very quick question, one very quick answer.
Well, I was going to say,
You got to ask it.
Jack instructed me to ask a short question. So in response to an earlier question, you talked about the combination of effects, the interactions, and last night we talked about chemical causes of asthma. Could you discuss the interactome?
No, can you not discuss it? Can you just make it through a short statement?
No.
Yes, and we'll have that in a minute, but Gary has to speak before we have another person coming.
So, and I think we may have time to discuss this a little bit more. I see these factors as selective pressures. Either they're antimicrobial or they select for organisms that can access those carbon sources and allow for them to have a fitness advantage, those microbes to have a fitness advantage in that environment. And understanding that interaction, I think, is really critical for us from a health status to understand the kind of the exposome and the totality of the exposome, both the structural factors that influence the microbes and then how those microbes interact in that environment and with the host.
Great. Gary, thank you very much.